Hello everyone, thank you for joining us. Welcome to our educational webinar titled Applications of the Hematopoietic CFU Assay in Toxicity Testing. My name is Catherine Lee and I am an Associate Program Manager at Stem Cell Technologies. I will be your moderator for this webinar. It is now my pleasure to introduce you to our speaker today. Dr. Jackie Damon is a Senior Advisor Hematopoiesis at Stem Cell Technologies. With over 15 years of experience in the company, as a senior scientist and director of contract assay services, she was responsible for overseeing client-directed customized study design and execution to evaluate the effects of both large and small molecules, drugs and cytokines on hematopoietic stem and progenitor cells for more than 150 biotech and pharmaceutical customers across the globe. Let me now hand you over to Jackie to begin the presentation.
Thanks, Catherine. Welcome to today's seminar, Applications of the Hematopoietic CFU Assay in Toxicity Testing. Today, we will be discussing how an in vitro assay using hematopoietic stem cells can be used in safety studies when testing for drug toxicity. To begin, I will review criteria to consider when developing an in vitro assay for predictive tox, and then discuss the process of hematopoiesis as well as hematotoxicity. I will cover the methodology of the CFU or colony-forming unit assay, and then finish by outlining applications of the CFU assay that can be exploited in drug screening. At the end, there will be a short question and answer period, so stay tuned for that at the end of the presentation. Okay, before I get into the biology of hematopoiesis, I think we are all aware and have tuned into this webinar because we know that testing drugs to ensure they are safe is an expensive part of the drug development process. Before a new drug can be approved, safety assessment involves testing for effects in vitro assays using immortalized cell lines, which are often tumor cell lines, as well as, and more importantly, in vivo testing using various animal models. There are problems, however, when using cell lines in animal models to predict toxicity in humans. Ideally, the reduction of the number of animals tested has been a mandate in research and drug testing for many years, and not only because animals are expensive, but because results obtained from these models is not always meaningful. Testing with cell lines, on the other hand, can theoretically help to reduce the number of animals required. However, the results from cell lines have been very deceiving, as we have come to realize over the years that these cells are genetically unstable, they often represent only a single mutation or mature cell lineage, and they are quite resistant to being killed in culture compared to normal adult cells. In addition, cell lines are often unable to perform the physiological function of normal mature cell types, so one can only measure simple endpoints, like whether they can grow or not in the presence of a drug. A good assay for predictive toxicity needs to be more relevant and approximate human physiology, so the use of primary derived human cells is really key. The ideal solution to better approximate toxic effects of drugs in vitro is to have normal cells from patients to develop functional assays so that effects on cell function can be measured directly. This has been a difficult problem in the past, as normal adult cells tend to grow poorly in culture, if they grow at all. In the last 20 years, however, we have learned that if we use stem cells, which are able to grow ex vivo and have the ability in the right conditions to differentiate and mature in culture to cells that are functionally identical to their mature adult cell counterparts, we find a much better correlation of in vitro safety studies with clinical trials. Today in this webinar, I will focus on adult-derived blood or hematopoietic stem cells and how these cells can be adapted for in vitro assays to screen novel drugs and drug combinations. Let's first begin with defining the criteria required to develop a good in vitro assay for predictive toxicology. There's been a lot of research in the last 10 years focusing on this area and the area of stem cells, both adult and primary stem cells, as well as embryonic-derived stem cells. Much of this is focused on the understanding and needs of these cells and the conditions required to induce stem cells to grow and differentiate into functionally mature cell types. The ability to generate induced pluripotent stem cells, or iPS cells, in culture, as well as iPS cells from diseased adult tissue, has caused a lot of excitement with regards to the ability to develop better assays, not only for predictive toxicity screening, but for screening drug targets specifically to disease states. In other words, the promise of stem cell biology is not only the ability to make cells and tissues that can be used in regenerative medicine, but also to generate physiologically relevant cells that can be used and adapted for in vitro cell-based assays. Having said that, before an in vitro assay can become a standard for safety and predictive tox testing, it must be shown to be robust and validated as a test that is representative of the biology of the cells that we are trying to understand. This slide highlights the criteria that came out of a 2008 workshop held by the California Institute of Regenerative Medicine on the use of stem cells in predictive toxicology. The participants of the workshop agreed that the use of stem cell-based assays in the process of drug discovery has the potential to reduce late-stage attrition, lowering the cost of drug development, and helping in the understanding of drug susceptibility. However, researchers in both drug discovery and environmental health realized that validation of these models is crucial before they can become standard for predictive toxicology. Outlined here are some key considerations for the use of stem cells in vitro-based assays that need to be met. Firstly, before an assay can be adopted as a standard, it must be validated by several independent lines of research to prove that it is physiologically relevant to the cell type under study. Secondly, that the stem cell derivatives used in the assay must be shown to recapitulate results found in other models. Thirdly, that the reference compounds with known human toxicity should be used for testing and validation purposes. Fourthly, that the methodology used to grow out functionally mature cells works consistently and is robust, that it is based on standardized protocols that can reproducibly give the same result with the same standard set of controls each time the assay is run. Lastly, and most importantly, one needs to show that the assay provides data which correlates to what is observed in vivo. In this webinar today, I will present data showing how one can use hematopoietic stem cells in an in vitro assay we call the CFU assay to predict toxicity of novel drugs and drug combinations for safety studies. This next slide outlines the process of hematopoiesis or blood development, which is unique in that it is the only system in the body and stem cell that is responsible for producing billions of cells every day. To better describe how this happens, this cartoon shows the hematopoietic hierarchy, which, on the extreme left, starts with the true hematopoietic stem cell. This hematopoietic stem cell is a cell that has the ability to replace the entire blood system in the donor for the rest of their life and is referred to as the long-term hematopoietic stem cell. This long-term hematopoietic stem cell is ultimately responsible for all the mature cell types found in the blood, all the red blood cells, white blood cells, platelets, as well as the lymphoid B and T immune cells. The hematopoietic stem cell is found in the bone marrow in a niche environment where signals from factors and cell-to-cell interactions with the environment ensures that there is always a good pool of these undifferentiated cells. Hematopoietic stem cell divisions are mostly asymmetric, resulting in a self-renewing division as well as a division that produces a cell that has a more limited potential and thus considered a progenitor. This very early progenitor is multipotent and is responsible for producing progeny for both lymphopoiesis and myelopoiesis. And when these progenitors divide, they differentiate into more lineage-committed progenitors based on the specific signals they receive from the environment. In this diagram, the progenitors are shown as cells that look morphologically identical, and it is these progenitor cells that are responsible for the constant regeneration of all the mature functioning cells found in the blood. The progenitors, as highlighted again on this slide, are referred to as colony-forming units based on their functional ability to proliferate in culture and fulfill their job of generating all the mature blood cells required in the blood, including the red blood cells from erythrocytes, the platelets from the megakaryocyte progenitors, and white blood cells, including granulocytes and monocytes and macrophages as depicted in this cartoon. Due to the enormous proliferative potential, these progenitors are susceptible to hematotoxicity, which is a problem and a life-threatening side effect of many drug classes, especially those that target excessive cell proliferation, which is a hallmark of tumor cell growth. This is the reason that chemotherapeutic drugs that target cell division also affect blood cells, resulting in complications caused by the reduction of different blood cell types. Anemia, for example, which is the reduction of red blood cells, neutropenia, which is the reduction of granulocytes, and thrombocytopenia, which is the reduction of platelets. Various drug classes have been shown to cause one or more of these cytopenias, and as a consequence, the dose of a chemotherapeutic drug used to treat cancer patients is often limited due to the hematotoxicity side effect, which can result, for example, in the reduction of neutrophils, leaving the patient susceptible to serious infections. Chemotherapeutics are not the only drugs, however, that cause hematotoxicity. In fact, there are several other drug classes, including antivirals, antimicrobials, drugs targeting tyrosine kinases, and topoisomerase inhibitors that have all been shown to have toxic effects on the development of blood with extended use. The CFU, or colony-forming unit assay, is an in vitro assay that utilizes primary hematopoietic stem and progenitor cells that are readily accessible from human blood, bone marrow, or cord blood, and can be grown outside of the body using the right nutrients and factors or proteins that we call cytokines. These factors stimulate both cell growth and maturation, and in the mid-80s, many researchers used the CFU assay to confirm the identity of these critical factors that, once isolated, could be shown to be important for the growth and maturation of very specific blood cell types. The CFU assay is an in vitro assay that has a very long history based on work started by James Till and Ernest McCullough in the 60s, when it was realized that you could measure the frequency of hematopoietic stem and progenitor cells by the fact that these cells, despite being present at very low frequencies in these blood sources, have the capacity to grow ex vivo in culture in the right conditions. The ability of these colonies to grow requires specialized semi-solid media, which specifically allows clonal growth of each hematopoietic stem and progenitor cell to grow in a spatially separated way so that they can be individually identified and scored as a way to measure the frequency of these cells in the body. As the first commercial supplier in 1993, Stem Cell Technologies focused on making a robust semi-solid media using methicellulose to grow hematopoietic stem and progenitor cells and provide researchers with a standardized tool that could allow them to quantify the frequencies of these important cells so that they could be evaluated and data compared across labs. A lot of research that started in the 90s has demonstrated that the CFU assay is a biologically relevant, quantitative, and therefore validatable assay with regards to predicting various in vitro parameters, including, for example, the ability to predict successful neutrophil recovery in patients following a bone marrow transplant. This assay is also required by the FDA for manufacturers of the cytokine drug EPO to document the appropriate biological activity of each manufactured lot before it can be used in the treatment of patients who cannot make their own EPO and therefore suffer from chronic anemia. Although a simple assay to set up, identification of hematopoietic progenitor cell colonies requires specific training. In this slide, I will demonstrate the steps in setting up a CFU assay. This first involves processing cells from either bone marrow, cord blood, or peripheral blood to first remove red blood cells before adding them to an appropriate methicult media containing a mix of optimal cytokines for the simultaneous growth of myeloid and erythroid progenitors. These cells and media are then mixed before the cells and media are plated into replicate 35 millimeter dishes or 6-well plates to ensure, and these plates swirled to ensure an even distribution of cells in the semi-solid media. The plated cells are then placed in a cell culture incubator for 14 days in the appropriate conditions for growth, that being 37 degrees, 5% CO2, and greater than 95% humidity, after which the cultures are removed and evaluated to determine the frequency of hematopoietic progenitors by counting the individual colonies and identifying, using morphological assessment, each colony type with the use of an inverted microscope. To evaluate the effects of small molecules or growth factors or drugs, these test articles can be introduced into the methocult media at the start of the setup for the CFU assay, before the cells are added, and then the effects can be observed by doing a colony count after the 14-day culture period. This slide shows images of an entire 35 millimeter dish to which 10,000 bone marrow cells were plated and cultured before imaging at day 14. CFU colony counts allow for a quantitative assessment and determination of the frequency of adult hematopoietic stem and progenitor cells, the number of colonies in the presence and absence of drug, as shown in this example on the slide, can be used to prepare dose response curves from which an inhibitory dose of the drug can be determined. In this example, you can see images from CFU cultures to which the same number of bone marrow cells has been added in each case, but with increasing concentrations of the chemotherapeutic drug doxorubicin. In the lower dose, 1 nanomolar, you can see a nice even lawn of colonies. The red ones represent erythroid progenitors, and the gray ones in this image, myeloid progenitors. And at 1 nanogram, the colony number and size are very similar to what is observed in control cultures to which no test article is added. However, as you see in this image, when you increase the concentration of the drug to 10 and 30 nanomolars, things change. There are fewer colonies, despite the addition of the same number of bone marrow hematopoietic stem and progenitor cells, and the colonies are smaller, thus demonstrating an inhibition on colony growth in the presence of doxorubicin. The colony readout can be used to determine the percent of colony growth for each colony type at each drug dose tested to generate a dose response curve that shows an inhibitory concentration, or IC value, where 50% of the number of colonies is inhibited. In this example, we can see that the IC50 values for inhibition of both myeloid and erythroid progenitors is similar, 26.6 nanomolar versus 21.4 nanomolar, respectively. As I mentioned earlier, a critical component for an in vitro assay to be used for drug screening and predictive tox is that the assay can be validated and shown to correlate with known effects of the drug in vivo. In early 2000, Stem Cell Technologies was approached by Dr. Augusto Pecina, a professor of general pathology at the School of Pharmacology at the University of Milan, who wanted help to develop a methocult media that could be used to evaluate the effects of drugs on progenitor growth in culture using the CFU-GM assay. We were, of course, happy to provide a customized formulation for his study, and this allowed Dr. Pecina and his colleagues to establish a protocol and validate the CFU-GM assay as a predictive model for myelosuppressive compounds. Studies by Dr. Pecina's group resulted in the publication of 2 key papers validating both the CFU-GM and the CFU-MK colony-forming cell assays as in vitro assays that can be used to predict the clinical effects of drugs in vivo. The first of these was the application of the CFU-GM assay to predict acute drug-induced neutropenia, an international blind study to validate a prediction model for the maximum tolerated dose of myelosuppressive xenobiotics, and this was published in 2003. This paper concluded that the CFU-GM assay could be used as a predictive model based on the results of 20 known neutropenic drugs that showed that the CFU-GM assay could correctly predict the maximum tolerated dose for 85% of the drugs evaluated. In fact, at a subsequent meeting held in March 2006, the members of the ECVAM Scientific Advisory Committee decided and endorsed the CFU-GM assay as an in vitro assay that could be used to determine safety doses of novel drugs before testing them in phase 1 clinical trials. The second paper was the application of the CFU-MK assay to predict potential thrombocytotoxicity of drugs. This was published in 2008, and this publication highlighted a relationship between the effects observed with the CFU-MK assay where they looked at the IC10, 50, and 90 values of 10 drugs known to cause thrombocytopenia, with the maximum plasma concentration, or Cmax, in patients above which thrombocytotoxicity was observed. The results again allowed the authors to conclude that the CFU-MK assay is a predictive model for drugs that cause thrombocytopenia. To highlight the robustness and reproducibility of the CFU-MK assay, this slide shows results that were generated within the Contract Assay Service Department at StemCell to test whether human bone marrow hematopoietic stem and progenitor cells would generate similar results to that published by Pacina's group, who actually used cord blood hematopoietic stem and progenitor cells in their studies. So as you can see in the results in this table, the IC50 and IC90 concentrations are either very similar to those determined in the Pacina publications, or at least have a similar molar ranking with regards to toxicity, confirming for us that human bone marrow hematopoietic stem and progenitor cells can also be used for in vitro testing of thrombocytotoxic drugs. A similar correlation was also observed for the CFU-GM assay, demonstrating that one can use hematopoietic-derived bone marrow stem cells to predict both thrombocytotoxicity and neutropenia, and testing drugs using adult bone marrow allows more flexibility with regards to evaluating the effects on different donors, donors of different ages, and also donors from bone marrow of patients with genetic disorders. Another advantage of the CFU assay is that it is customizable and flexible. As shown in this diagram, specific hematopoietic progenitor subtypes can be evaluated by changing the culture conditions and cytokines. Since the growth requirements for different mature blood cell types have been identified, one can look at more specific effects of compounds on neutrophils, for example, compared to effects on monocytes and macrophages by just changing the growth conditions and the cytokines. The data on this slide, for example, shows that different tyrosine kinase inhibitors affect these mature cells differently, as shown by the IC50 values for imatinib and sunitinib on the number of CFU-G progenitors versus the CFU-M progenitors. This type of analysis and assay design highlights how one can use the CFU assay to help identify efficacy as well as toxicity of drug targets on specific mature cell types. The versatility of the CFU assay is another advantage for predictive toxicology, since it can be performed using bone marrow from various animal species. This slide highlights results of the CFU-GM assay performed using bone marrow from mice, rats, dogs, and humans for 10 compounds from 4 different drug classes. From this particular example, the results clearly show that the IC50 results can help determine which species is the most applicable as a model system for in vivo studies. In this table, you can see IC50 values that were generated for 3 main classes of drugs: topoisomerase inhibitors, chemotherapeutics or antiproliferative drugs, and tyrosine kinase inhibitors, as well as 1 known environmental toxin. Originally, we set out to perform these experiments on topoisomerase inhibitors, as there was evidence in the literature that dogs were a better model system than mice or rats for this drug class, so we tested this in-house using the CFU-GM assay. As you can see in this table, if we focus on the results for the 3 topoisomerase inhibitors, we can see that the IC50 values, based on the average of 4 independent experiments, with human CFU-GM in the first column versus canine CFU-GM, you can see that these are almost identical. When you compare these IC50s to what we saw using mouse or rat, you can see that in rodents, the IC50 values were found to be much higher, and in some cases more than 10-fold higher for mouse CFU-GM than observed with human and canine CFU-GM. This is opposite to what we found for 3 antiproliferative drugs, where you can see from the data here that the IC50 values for the human CFU-GM were almost identical to that observed for the mouse CFU-GM, versus what we observed for canine, which was found to be much more sensitive to these drugs. Interestingly, for the drug kinase drug class, we found that the canine and human IC50s were again more similar and very different when compared to the IC50 values observed for mouse. especially in the case of the drug imatinib. In summary, I think it's fair to say the results here show that the CFU-GM assay can be used to help determine an appropriate dose before initiating in vivo testing in various animal models, and it can also be used to evaluate the appropriate animal model that correlates best with the human response. To further highlight the versatility of the CFU assay, this next slide shows the results of testing drugs alone as well as in combination with other drugs. Before treating patients using drug combinations, one can therefore use the CFU assay to determine if the toxic effects of adding two drugs is expected to be just additive, or if the particular drug combo acts synergistically, resulting in much more toxicity than expected. In this example, test article 1 shown here is tested alone and in combination with IC20 doses of 3 known chemotherapeutics, cisplatin, TMZ, and SN38. In combination with cisplatin and TMZ, the toxicity of test article 1 is seen to be additive. However, addition of test article 1 with SN38 shows a significant shift, indicating that test article 1 and SN38 actually have a synergistic effect. This type of analysis can therefore confirm not only the efficacy of drug targets, but it can also provide important information about synergistic toxic effects before initiating safety studies in patients. One last application of the CFU assay that I will highlight today is the ability to use this assay in the assessment of various blood disorders and diseases like polycythemia vera and various leukemias. Historically, hematopathologists have used the CFU assay as part of their diagnostic criteria for various blood diseases. More recently, as seen in these studies referenced on this slide, the CFU assay can also be used to test drugs directly on diseased patient samples. So far, we have been talking about the CFU assay as an in vitro assay, adding drugs directly into the cultures to evaluate effects on the growth and maturation of the different blood lineages. However, the CFU assay can also be used to evaluate the effects of drugs in vivo on actual patients with diseases like leukemias and myelodysplastic syndromes, where the CFU colony growth is typically abnormal. For example, during clinical trials, when one is looking at the efficacy of treatment, samples can be drawn from the bone marrow or the peripheral blood from patients before and at various times post-treatment to evaluate if there are any improvements in the functional ability of progenitors to grow out as a consequence of the treatment. In summary, I hope I have outlined in the webinar today how the hematopoietic CFU assay meets the criteria of an in vitro assay for predictive toxicology, based in part on a body of literature spanning more than 20 years that has proven that this assay can be used to measure the frequency and functional ability of blood progenitors, as well as the fact that it has been validated to be predictive of the in vivo results. The power of the CFU assay, of course, is that it uses patient-derived hematopoietic stem and progenitor cells instead of cell lines, and these are more physiologically relevant as well as quantifiable. I've also shown you results that the CFU assay is flexible and has the potential to be customized to examine the effects on very specific blood lineages and specific pathways, making it an assay that can also be used to determine synergistic effects of drugs and provide information on the mechanism and potentially the efficacy of drug targets. This concludes this webinar for today, and please stay tuned for our next webinar that will discuss more high-throughput methods for screening hematotoxicity that can also be adapted for hematopoietic stem and progenitor cells from normal and diseased patients. I will now open the floor for questions. Thank you for your time and attention.
Thank you very much, Jackie. That concludes the presentation segment of the webinar. We will now begin our Q&A session. Can you differentiate between the effects of drugs on cell growth versus those that inhibit cell maturation?
Good question. Yes, depending on how you do your drug and when you incubate the cells with your drug, you can get different kinds of information. So in some cases, and depending on what you were looking for, it is possible to take hematopoietic stem and progenitor cells and put them in sort of a 24-hour or even a 4 to 6 hour, what we call pulse, that allows you to have the drug interact with the cells. And if you do that for various time periods, so if you look at a 4, 6, 12, and 24-hour time points, for example, which is typically the time it takes for cells to proliferate, in this case, you can get a really good idea if the drug is actually affecting proliferation. Because if you find that you don't have an inhibition at 4 hours, but you start to see an inhibition after overnight, and that actually gets, you get more inhibition after 48 hours, that can be observed by taking the cells after those pulse time points, washing out the drug, and then putting them into a CFU assay versus a control that isn't pulsed with the drug to show whether there's any differences in the output of CFU from sort of the same number of cells put in because of the effects of the drug before you add it into the CFU assay. So the reality is, within the CFU assay itself, you can't tell the difference per se because the cells are dividing and maturing every time they divide. But if you want to start to differentiate and distinguish these two, you can at least evaluate proliferation by looking at putting the cells for a shorter period of time and in different time courses before you wash them and put them in the CFU assay as a readout to see what the effects were on those progenitors.
What is the approximate quantity of candidate compound required to generate a dose-response curve with the CFU assay?
Another good question and a very common one. We actually realize that most drugs are in the toxic level of micromolar. So that's where a lot of these kinds of assays, that's sort of the range that's used. Once it gets down to nanomolar range, then you know your drug is really toxic. So typically we test at starting with either 150 or 30 or 10 micromolar and then work down from there, assuming that that correlates somewhat with what's going on in vivo and sort of the maximum tolerated dose of what you see these things, if you've got any historical data and any in vivo data. But you start with usually, like I say, about 30 micromolar and work your way down to 30, 10, 0.3, 0.1, and look for where you have eventually lose that inhibition so that you can generate that dose response curve. Having said that, if you are in the micromolar range, then you are going to have to, we would need compounds in the millimolar range, and typically 1 in a thousand-fold stock of what your highest test concentration is. So if you want to test 10 micromolars, we would need to have 10 millimolars, but we don't need very much of it. So the assay in itself is only done in replicates with 1 mil. So having 50 to 100 microliters of a thousand-fold stock of the highest test concentration is actually all you need to set up a good dose response curve using the CFU assay.
Is it possible to test compounds for effects on lymphopoiesis using the CFU assay?
Yes, I wish that were true. Lymphopoiesis is more complicated. You need different environments. So you have to think of lymphopoiesis more like, for anybody that works on pluripotent stem cells, you need the ability to take these cells through different phases of their development. Erythroid and myeloid actually can occur quite easily just from the progenitor stage by providing the right cytokines and serum conditions, whereas something like B and T cells, they actually only have a few divisions in the bone marrow and only become sort of a really early progenitor type before they move out of that environment into the spleen and the thymus, where they sort of continue that maturation process. So we don't think of that in the same way, and the conditions for that are actually quite complicated. Historically, that has been, to look at lymphopoiesis, you have to do co-culture, although things are changing in that arena now, and people are using sort of trying to mimic what happens in those other environments like the thymus and the spleen by coating culture wear with different peptides and proteins. So that's changing, but at this point we don't actually have a similar, what we call CFU assay for T or B cells, with one exception. We are able to grow B cells in a more mature state out of mouse bone marrow, just adding one cytokine, for example, which is IL-7, and that alone allows you to, in mice anyway, produce a lot of divisions from that progenitor and a colony that you can identify. Unfortunately, with humans, that is a lot more complicated process, and there's a lot more involved. So it's not that simple, and they do not form under simple conditions an actual CFU. So those assays are usually done in mouse models, as well as in some cases long-term bone marrow culture models.
Is there a drug type that could show a false negative in the CFU assay?
Good one. Yes, I think the only time we've really ever seen that, having said that, is if there's solubility issues. So if you find that you have a high test concentration of your drug and everything looks fine and the colonies look great, and then as you look at decreasing concentrations, all of a sudden you see no colonies, that's usually indicative that at that highest concentration where the colonies looked okay, that the drug came out of solution. And usually you can see that and visualize that when you score these colonies by the fact that you'll see crystals in the media. So that's sort of a telltale sign that the drug concentration is too high. And it can happen because there's a lot of things in that media. There's a lot of salts, there's a lot of serum. So there's a lot of things that could cause these drugs to precipitate. So good solubility at 1 in 1,000-fold less stock from the stock that you provide, like a 10 micromolar, is really important to know before setting up that assay.
Okay, I think that's all the time that we have for today. I really want to thank everyone for attending, and I'd especially like to thank you, Jackie, for your time and an excellent presentation. Thanks again, and goodbye.